So we are live now. Hello and Assalamu alaikum everyone. My name is Arsalan Riaz and I am the Vice President of RSG Pakistan. Welcome to the second episode of Computational Biology webinar series organized by RSG Pakistan in collaboration with Bioinformatics. Today our speaker is Dr. Sundar, who is a Ph.D. in computer science from the National University of Singapore, and also he has been associated with Bioinformatics as an advisor for a long time. With Dr. Sundar, Dr. Prashant has also joined us, who is the current leader of Bioinformatics, and he will be giving us a little brief about what Bioinformatics is. So on to you, Prashant.
Yeah, thank you very much for this kind introduction, Arsalan. Congratulations on organizing this, and it's been a great initiative. So I'm Prash. I'm the founder of BioClues, and as well as a senior scientist at Birla Institute of Scientific Research in Jaipur. So BioClues stands for Bioinformatics Club for Experimenting Scientists, which I founded it in 2005 along with Pritish Bharadwaj. So it went on to become an affiliate of International Society for Computational Biology in 2009. And then we have mustered the support of Asia Pacific Bioinformatics Network, where Asif M. Khan is the current president of AP Bionet. So over the last 16 years, we went on to become India's largest bioinformatics society, and we have mentored more than 4,000 graduate students who are placed across at least 30 different countries all over the world. So our main forte is to work towards mentor-mentee relationships and ensure that all bioinformaticists come together towards pinnacle of success. So this is a very brief thing about BioClues. But today's guest is Dr. Sundar Rajan, Vijay Raghava Seshadri Sundar Rajan, also known as Sundar Da in brief. He has been my mentor since 2010, and I've greatly worked with him on various aspects of it, especially on development of machine learning based approaches for hypothetical protein prediction. He is currently a senior data scientist based in Singapore. Besides honing as an entrepreneurship chair for BioClues, he has also been one of the chief advisors for BioClues since 2013. Apart from that, he started his Ph.D. in computer science, obtained his Ph.D. from SANBI, Cape Town, and later on he went on to gain several years of postdoctoral years of experience. As a part of his PhD, developed one of the well-known antimicrobial databases, which was featured as one of the best database papers in Nucleic Acids Research Journal, published by Oxford Publishers. And then even before that, he has had two decades of experience working for prestigious Bose Institute in Kolkata. And then he was working for A-Star Singapore and S-Star Singapore. And then he was piggybacking and mentoring as many as several graduate students working across different spheres. His main areas of expertise is coding. As you could see, at least the 11 or 12 databases that he has produced, there are huge lines of codes that he has marvelously developed and as well as indexed. And they're all not just databases, they're all reinstantiable codes that he has wonderfully developed for the research community. And Arsalan has been kind enough to draw us together. And this is why Sundar Da is here today. So I'll not bore you all, with all these kind of things, and let's wait for Sundar Da's presentation to kick off. Thank you. Thank you, Sundar Da. Please go ahead.
Yeah. Thanks, Arsalan, and thanks, Prash, for a detailed introduction. So the idea is data science on one side. The idea on the other side is bioinformatics. Many of you are going through various courses in bioinformatics hoping to get something out of it. This is how most of us started. Even when I started the bioinformatics almost 30 years back, without knowing how it will going to go. It has graduated in a very detailed way, not only in biology, chemistry, and other domains. It also went through for banking, insurance, and where not. Many of you may ask a question, where these bioinformatics things are going on. Then the tools of bioinformatics are immense, and that proven a lot of things in biology where it is otherwise difficult to do an experimental way of getting things. So
Sundar,
let us.
sorry for interrupting. I think we cannot see your slides.
Yeah, I'm going to the slides now.
Okay, sorry.
Can you see the slides now?
Yes, we can.
Okay. So today we are give you an overview on what is the big data now coming up and what it has impact in bioinformatics. And also I want to caution people who jump into say that I want to do big data. A lot of data is available with me. And also we'll show how we are going to use the big data in bioinformatics or related field and how it is going to be useful if we go in a structured way rather than getting everything onto us. Thanks for RSG Pakistan for organizing this seminar. So we will go on this pattern. How we all know the massive data is derived and how the cost is fluctuating due to the advanced technology. And on the other hand, we need to do a proper disease modeling because like COVID, we are now working on the COVID database with school children to see whether we are able to do some modeling to make some idea how the things are happening on the COVID front. And then we will see how the bioinformatics and big data has evolved and how things are happening and what we can do with the data and analytics once we have the data. So we should have a blueprint what we are going to do with the data because data is available. We need to know what we want and what we want to solve, what we want to structure, how it is going to help in using these ideas. Then we can do a better way of handling things rather than we get huge amount of money in doing only experimental analytics rather than silico experimental analytics. That gives a lot of goodwill that we are going to see in this lecture. And what are the impacts and then what are the challenges because I simply say big data means I can do anything. Even today there are a lot of challenges and I will just show two paper as Prashant mentioned about the nucleic acid. This is just a few minutes I will show and finally what we are developing on the Sol corona database. Okay, let us start. We have huge amount of data, mostly data, and most of you know that whether I call it NGS, database, or FASTA, homology, any word in this slide will tell that that brings a lot of huge data. The only thing is we are worried about how we store it, how we transfer, how we analyze. Can we do some visualization on using our data which we retrieve from online resources which are freely available based on our storage capacity or transfer or Wi-Fi or network capability. We are able to get data and then what we are going to achieve. So as if data is already there and we have to see what we are going to do with it. And also the cost of genome has drastically come down. As per the recent thing, the per genome, the cost is so less that we are able to do more of getting genomes. Now, every lab is ready to prepare a genome sequence with the thing which they want to do. So, as it is declining the cost, it is possible that we are getting more and more data. But one question always there: how much this data are accurate? It's always a challenging question, which lab has no answer. But in silico, we are able to see whether this data has some problem that we are unable to use it or what. That's where our bioinformatic tools are handy and helpful and solve these issues. So the major question the doctor and scientific community is willing to ask one question: what causes the genetic disease like corona or any disease? Can we map it? Is it possible for us to find how things are happening? And is it possible from human to human, if we can make a genome sequence in future, then is it possible that we know how the diseases are spreading, what causes the disease and things like that? These questions are always pending now. So that is the idea we are going to do. One side is the genomic information is huge. On the other side, we have a disease problem. So can we handle this? So now, after all this scenario, we are coming to the listing. What are the bioinformatics and big data? So bioinformatics is one side, is the basic data analytic tool. It is having a lot of potential tools in prediction, modeling, or how we are able to handle things. On the other hand, we have a volume of data. It is coming from various sources, either it is from clinical or genomic lab or protein structure or even NGS analysis. So many ways we are able to get data for a protein or gene or things. Now, what we want to do with this data, either identification and treatment of a disease at a faster scale. So if we can pinpoint genomic level or which causes the thing, root cause, then we are able to find the disease as quickly as possible, and it will decrease the number of people getting infected or treated. Even like cancer, still we are having a miracle in solving it. We could solve that particular cancer problem, even though it is not yet solved. We are able to get the patient at the level 1, degree level 1, rather than until they go degree 4, then it is then only it is diagnosed, and it is becoming difficult to save the people and also reduce the cost. So instead of going so many labs, if we can 50-70% cover through the silico, then the cost is reduced in analyzing anything in disease model or either genomic sequence, and then we are able to provide better health at the early stage, rather than we say a person going to die soon. Rather, we can prolong a particular life, because if we can predict early the disease. And care of patient means regular data. Once we get, then we are able to moderate it and amend how the treatment adjustment, and then we are able to make people more happy and they're good in shape. And disease control is a very important thing, because how things are spreading like COVID, we don't know what is happening behind the scene. So all these population disease things, if we can model it in a proper way, then we are able to control it also. So what type of data is collected? As we mentioned, DNA sequence or amino acid, protein structure, 3-dimensional, and etc. So the bioinformatics is an explosive growth starting in mid-90s because of the Human Genome Project, with a lot of advances in technology. So what is the analytics on the other end? It is able to solve some issues, like whether the gene can be predicted for a protein function, or we can predict partial of the sequence, or how we are able to bridge whether this group of genes belong to a particular group of function and structure, everything. We are able to now build such things. How we are doing? We get the data, what we know and what we don't know, and with that we are able to do a mathematical algorithmic model that helps in making the model fine-tuned, so that we are able to predict as much as possible. That really reduces the cost. Okay, let us now in detail see what are the impact of big data in bioinformatics. Let us go by topic. Let us go biology. The traditional way, what he used to do, we go by the traditional way of finding things. But now, due to technology and silico-based methods, the big data has helped a lot biology in making it time-consuming, less of time, cost-effective, and we are able to do better analytics direction. If we know that particular gene, instead of experimental, we know where it path to, which path it is going to be. Then we concentrate on those things so that we are able to match the protein function as early as possible with less cost and less time and things like that, rather than searching it in a big ocean, which is very difficult to finally get. So we are able to predict protein structure and also the gene sequence. We are able to, usually we build data, gene sequences, the laboratory and things like that. And then we map it to genes and identification of a lot of things, genetic disorder. All these are traditionally done. Due to big data availability, we are able to match them and get the hidden info so that we are able to reproduce some of the part of the sequence because of the, even in COVID, which we are analyzing, lot of sequence is partially the same. So only some mutation is happening. So that helps instead of getting a new sequence. If we can get partial sequence, then only the mutation portion we are able to bridge it. Then we are getting the sequence without building it from scratch. And main thing observed is laboratory may have errors, but analytical tools, they don't produce errors because once things are clear to the computer and do the algorithmic methodology, then we get answer is more accurate than the traditional way. Traditional way can have any mistake like probe problem, dust in something, or the methodology, some human error, anything can happen. But computer, nothing can happen. When it works 100%. So there is no chance of error, and the outcome is more accurate. Healthcare, as I mentioned, a huge collection of diseases. We are also trying to collect various details so that we are able to monitor the health and big diseases like diabetes, heart diseases, cancer condition. All these can be mapped, and we use the bioinformatic technique. That is the base to understand how things are happening genetically or medically and things like that. And we are able to correlate between genomic and medical. Doctor see only the medical portion, and we see the genomic. And bioinformatics helps in mapping them so that we are able to go close to the point how things are happening. Faster identification of proper treatment that obviously reduce treatment methodology, better health and patient care is obvious. We are able to get things hand in before rather than so many times the doctor saying the medicine and finally comes to a level by the time the disease has increased in a very advanced level, it becomes very difficult to treat properly and enhance the patient health and life. So, and many people are aiming on this preventive and personalized medicine, even though it is slowly coming up, but still not in action. Like a person can carry his genome on his hand, with a, print something like that, then he can go and see doctor, and doctor can prescribe personalized medicine with the use of genomics, proteomics, metabolomics. Then he gets a proper potential drugs. When manufacturers can produce the drugs with a composition needed for that patient, depending on the his problem level, disease level, rather than as a common drug for everyone. Even though the person starting from 0 to 10, they get the same medicine. Maybe the doctor take one time or two times. Like it is increasing the number rather than it is not adjusted for a personalized medicine. These things are expected in near future, so that we are able to get everything done in silico, and we are able to prevent diseases, disability, and death, and things like that. So What is the impact on deep learning and machine learning? Machine learning, as some of you may be knowing, is how the machine learn things from the data I give. For example, if I take gene and protein function, if I give list of genes which are going to do the protein function and give which are the genes which are not, I don't know the function, then machine learning models, maybe in a future lecture we will be describing and demonstrating how it is happening. That helps in getting an inherent so that we are able to know this set of genes, how it is going for the protein function. So on biomedical data also, machine learning does a lot of work. We are able to model diseases, and we are able to do predictive and prescriptive analytics. And what is deep learning? If I know one particular type of cancer, some data I have enough data, but I don't have data on another type of cancer. So how I model that type B, even though I have a lot of data? Data means I have the patient detail, those who are having that type A cancer, and also their characteristic and assays, pathological report, X-rays, everything. For other set, I don't have control set. Mean it's not much known, but I have the data, general data. So how is it possible for me to model this? So there comes the deep learning that can transfer that knowledge learned from type A cancer to type B cancer and even an inherent of how it is grouped between cancer yes or no. So that gives an idea, and then that brings an idea how we are able to make further layer and build the model in a model's easy way. The type B, which we didn't have proper data with us. So machine learning always we need yes and no. Yes means I know the result with this data, whatever data, and no means I have the data. I don't know whether cancer exists or not. So that the machine learning is able to build things. And deep learning helps further. After I build model for type A, I can put it for type B also. And that gives a inherent in dividing them between yes and no. That gives more better idea. And this we can go for a cloud platform, cloud framework tools where you can pay as you use, and this all give a better understanding of health and disease. This one I will share later. You can go through some of the thing happening in big data in informatics. What are the big data? All this. Okay. So far we have seen everything looks very green and nice, but we have challenges. That's what I was mentioning in the beginning. Things are not so easy, and sometimes it becomes harassment also or frustration and things like that. That's what we also face day to day, and finally we manage it and solve it or resolve it. So when business analytics, when we are unable to model anything, whatever be the field, biology or doctor or disease or anything, why it happens? Because lack of data. Because the data we assume with us maybe not enough or biased data. Because some people don't give proper information, like even gender or age group or a lot of things. We have seen when the people fill up the survey, they don't fill up a lot of things, and only some data we have. So that becomes difficult to know whether it is spreading in population or religion or region. That becomes very difficult to find things in an accurate way. And also sometimes we don't get data updates very easily. Whatever we have known cases, with that we have to struggle. We don't get the new data, proven data, so that our model becomes more good in making things better. Traditional doesn't match in the new model because whatever we did in the traditional way of understanding things, it doesn't happen in the new model as lack of data. And if we cannot build the proper model, the prediction becomes very difficult and it becomes not even 50% level. Then we cannot say whether this model is good enough. And expert knowledge is sometimes not available. Even the modeling, we always take expert knowledge, whether it's a doctor or surgeon. so that he has more idea, and we can always discuss with him to make the model fine-tuned so that it can be more good and better. If we don't get this knowledge input to the model, then it becomes very difficult to fine-tune. Regarding the accuracy of analytics, sometimes poor quality of data. Even when I started my PhD, I used a lot of data, microarray data, but most of them didn't do anything good because of the problem in the data, how it is done. Even though it is done in a one go of 7,000 genes or like that, but the quality is not that good. So that becomes that data is not usable, or it is not having the same accuracy. If I use different microarray, it doesn't fall into the same way. Like I cannot model it for a set of protein function, and not consistent also, because every time it changes, because the data is not clean or a problem. Even in disease model, it cannot be balanced. We cannot say whether the disease exists or not, because the data is not that accurate. So data analytics become more complicated. Why? Because we cannot have a clear visualization, or the model is more biased, and we cannot validate properly, or the data is not that useful. So involving external is a problem, and the suggestions are bring external domain into build the model with subdata. Means can we divide the data as a subset and try to build a modeling? That this we have done in dentistry, and we found that gives a better way of getting model rather than taking all the entire data into the system. So this is all suggestion. And when we take more data, what else we can have? The system can crash. That happened to me also many times, because we think we accumulate everything and run to the computer. Computer has a limitation of memory, and so anytime the system can crash while getting the model. And time delay. Also, the system may delay things, and finally we don't know after some time whether we are able to get a crash or getting a knowledge. The volume of data is not in order. So we can improve it still, the improvement of infrastructure and resource utilization, or we can use a depth data set, or these are all the scaling is a new way of stepping up data. Use a first set of data, then use another sub data. Like that we can upscale things so that we are able to build a system without going in for a crash using all the data. Finally, when you have huge data, what are the major issues? Expensive maintenance. Need a huge storage requirement, and that becomes a more cost. And you need to always plan for a better hardware. So the good suggestion is go for cloud solution, where you can pay as you use. When you use more, you pay more. When you don't use more data or memory, you pay very less. So that brings reduction in cost, and simpler version, and then scale up system. So these are the blue or the suggestions. So how we are able to get all these problems in mind, so before we decide what we want to do, then we can plan accordingly how we use the big data in getting to what we want to analyze, what we want to build, or what we want to find from this data. So I want to show two case studies. I think almost we are 30 minutes, right? So this is one of the paper which was mentioned by Prashant also. Even this one, I am expecting some research scholars who are interested to release the new version for this. If anybody interested can contact me, because this will take time. You can publish it again with the new updated data, but it takes time, at least 6 months to 1 year. Some group has to work on it. So this one is antimicrobial peptide database. We build it way back in 2010 and published in 11. And this one is, so from the big data, we are able to get the data according to a particular type, like antimicrobial peptide. And then we build this data after do a lot of creation, and finally we found out of several thousand, only about 1,000 plus we selected. It's a very good, useful resource. And yeah, this one I always like to quote that how we do on one side, big data, we are able to do a database level, which is going to be more useful for those researchers who want to get some data and analyze that sequence, again, blast it against the database so that they get all the information before they want to do anything else. Similarly, we are building a next one. We are building this COVID database. This one you will be surprised that we are using school children to build this, and they are very curious and they are building this. Very soon we will publish this. You can check on the website given below. It's not completed, still it's work going on. And so here also we know many labs are producing sequences. So they can see whether it is a new strain of a COVID or not. How they can find, they can come to this database system and blast the sequence against the available database here, sequences in this database. So it will give an idea whether the new strain is how much is different from the different strains found in the database, and that helps in getting a quicker and early information. Okay, any question? Big data for this.
Thank you so much, Sundar, for our wonderful webinar. So we have some questions. Okay. The first one is why DAMPD database is down.
Yeah, it's a good question. The thing is, that's a problem of informatics, right? When I go for a postdoc, so I had a server, I implemented it, and everything was fine. When I left it, the server down, and no one is there to take care of it. So this is a problem happens in the field. It's also good. If another researcher want to build, we can build it, rebuild the database and make it alive in another place. So that is possible.
Okay, so I think that we don't have any more questions. Thank you, Prash. Thank you, Dr. Sundar, for a wonderful webinar. Um, I think we will end now. If there are any final notes from you or Prash, then we can
Oh,
go.
thank you, excellent for organizing this. Thank you so much. Yeah.
Thank you so much. Thank you so much,
Yeah,
Mr.
thank
Sundar.
you. Yeah, thank you. And we welcome people to work on with us. We can rebuild things in a better way. We are there always. We love to have more students from Pakistan or anywhere else. That's what they are striving, and we are always carrying through. Okay, thank you.
Well, actually, in the comment section, we have someone that he's interested in a postdoc position in computational analysis of plants and pathogens. Do you want to say something about that?
Actually, I told Prashant told no, I'm not working in a lab or a research institution now. I'm working for government actually. So obviously we don't have this position. They can search actually. Postdoc position they can search online everywhere, wherever whichever the country, and they can apply. The outside is if you are qualified, you are proof with good work, they will call you definitely. It's not difficult going anywhere.
Okay, so that's
Just
it
to
from our.
add to this point, excellent.
Yeah.
Adding to Sundar's point, a postdoctoral application is actually a kind of an art. Actually, So many budding PhD fellows from South Asia Pacific regions, we usually don't move to that particular stage that easily in Europe and US. That's because we probably end up writing a very poor covering letter or a poor CV. So for this, we have managed, to identify, making a good course on effective cover letter and as well as CV. So we through BioClues, keep organizing this at least, once in 6 months. We have been doing it. Nevertheless, what he could do is he has to write a good covering letter in no more than one page, keeping his CV, that too in not more than again 2 pages. And please ensure that the cover letter is not more than 3 paragraphs. The first paragraph should describe about himself or herself, why he is really interested in that particular position. The second paragraph about his past background, and third one is if there are any inherent expectations for that particular postdoc project, and finally ending with a small statement on the family as well. Because the European way of looking at such kind of things is they always try to look how best we could really help this particular candidate achieve this particular position. Is he along with his family or is he along with his friends to be supported with? So these are the kind of questions that the Westerners look at it. So unless and until you end that particular statement, probably your CV or your particular application is not considered at all. So this is about science. So these are all the nitty-gritties that I could suggest the candidate. In any case, just keep on applying at least in a day. At least you should apply 2 or 3. Yeah, all the best. Yeah.
Thank you so much for the great advice. I think Adnan Ahmed may found it helpful. So thank you so much once again for joining us and having a webinar with us. Thank you so much. We will see you in the next one.
Thank you. Bye
Bye
bye.
bye. Allah
Yeah,
Hafiz.
all of you. Thank you, Sundar. Thank you,
So
Arslan. Yeah.
Thank you.